Okay, so I'm going to talk about the 5 different kinds of vaccines and how they relate to SARS-CoV-2 vaccines that are being developed currently. So here are the 5 that have the 5 types of vaccines. The virus vectored vaccine, so basically that's engineering a virus, it's usually engineered cold virus that can carry the DNA that will code for some protein on the outside of SARS-CoV-2, spike protein in general. There's the attenuated viruses. Now generally these are live viruses or infectious viruses that generally when you make them, you have to pass the bad virus through a bunch of cultured cells over and over and over and over again, and then artificially select for virus that isn't likely to cause disease. So this generally takes a lot of time. So when people usually say that vaccines usually take 5 to 10 years to develop, they're mainly talking about this type of vaccine. So we don't have any in clinical trials yet that are attenuated or quote unquote live. Viruses aren't really alive, so that's why it's weird. There's heat-killed virus or inactivated virus. There's 2 vaccines, both Chinese vaccines, that are in development. So these aren't live, or they're not infectious. And then there's nucleic acid vaccines. This is like the mRNA vaccines. There's 2 US vaccines, Pfizer and Moderna, and I'll explain how those work. And then the subunit vaccines, which are probably the easiest to produce. Oh, these are both pretty easy to produce. Which there's a US company called Novavax that's making a subunit vaccine. So I'm going to go through each one of these and kind of explain how they are created and how they're different. And then I'll just refer back to which vaccines I'm talking about, what companies. So first I'm going to talk about the virus vectored vaccine. Now I have another video that's a lot more detailed about this, but for bioethics you don't need to know all the details. So this vaccine was originally called ChAdOx, and it stands for chimpanzee adenovirus, Oxford University. But they changed that to this really catchy name called AZD1222. So this was developed by Oxford University, and one of the reasons why this vaccine is kind of further along than other vaccines is because they had already worked on this for MERS, which is the Middle Eastern version of SARS that was several years ago. And so they'd already gone through a lot of the process with essentially the same prototype, but with an inserted gene that was more specific to MERS. So they got a head start when they just switched it out to being SARS-CoV-2. So basically you take a virus. Now this is a cold virus. Adenovirus or adenovirus is a cold virus. It's a lot of different viruses cause the common cold, but one of them is adenovirus. Now you don't generally want to use a human adenovirus as a vector because we already have immunity to those. And so if you injected somebody with human adenovirus, generally you're just going to attack the vaccine and it's not going to be able to do anything. So they use a chimpanzee adenovirus. And what they do is inside the virus is its DNA. So this is a DNA virus. And they engineer the DNA in the virus so that it can't reproduce in cells. The point of it is not to get the cold. The point of it is to use the cold virus as a way of shuttling in the gene for the spike protein. So you engineer the heck out of this. It's called recombinering, where you just basically cut and paste genes out of the normal cold virus, and you put in genes. You put in a gene for the spike protein for SARS-CoV-2 into the adenovirus genome, and you take out genes that would be necessary for it to reproduce inside cells. So it's altered in that it's only a vehicle for bringing in a gene. It's not really a vehicle for copying that gene. So once the virus gets inside of a cell and it goes into CAR positive cells, So different cells then would be infected by SARS-CoV-2. But they go into these cells, and then the gene gets transcribed, and those cells produce spike protein. And those spike proteins are then released out into the bloodstream, and then your body will launch immune response to the spike protein. So it's basically a tricky way of getting you to make the spike protein so that you can create lasting immunity to that. So the slightly creepy thing about this type of vaccine is it's basically, first of all, it's genetically engineered, which a lot of people just have trouble with anything genetically engineered. And second of all, it's actually causing some of your cells to become genetically engineered because you get the gene inside your cells for the viral spike protein. Okay, so that's the viral vectored vaccine. Now, if we look again, that's the ChAdOx, or the AZD1222, created by Oxford University. This is in phase 3 trials. Already in the UK, there's, I don't know, something like 50,000 people that have already been vaccinated. The placebo, they decided to, instead of just giving them salt water, they're giving them a different vaccine. So it's like some kind of, I think it's a meningitis vaccine. So they still get, the placebo people still get some of the side effects of getting a vaccine. Like they might get a slight fever, or they might get arm soreness, so they won't know if they got the real thing or not. That's kind of, I thought that was kind of slick. Okay, again, attenuated virus. I'll show you what that would be like. An attenuated virus, so this is my model that looks a little bit like SARS-CoV-2 in that you have an envelope, you have spike proteins, and you have RNA inside. So an attenuated virus would be a similar virus to SARS-CoV-2, except that it doesn't cause disease. So you have to, like I said, you have to pass it through a bunch of human cells in culture and show that it is weakened or can't produce disease. But the advantage of having an attenuated virus is it tends to give you a more robust immune response because it's a lot more like the real thing. And it's infectious. It actually goes into your cells and reproduces to some extent, but it doesn't cause disease. So that's an attenuated virus. Again, there are no clinical trials right now because this takes longer to develop as of now, as of August 2020. Okay, so heat-killed virus or inactivated virus. Actually, I'll just use this model. It would just basically, you just take SARS-CoV-2 and just destroy it so that it can't be infectious. And then you just use that. And this is like the old school, the way they used to make vaccines when they first started making vaccines. They just boil it or break it up in some way. So you still get the proteins that are on it. And you'd get more than just the spike protein. So you'd have more places where you could make antibodies, but it's just simply destroyed virus. And so there are 2 Chinese companies, Sinovac and Sinopharm, that are making heat-killed or inactivated virus. A lot of virologists think that this is going to be a really good candidate because they're worried that if we all only focus on the spike protein for our vaccines, that if that's not how we get immunity, then we're going to miss the mark. So with this kind of vaccine, or an attenuated vaccine, you would get more of a broad range of antibody responses. And more importantly, what we're finding out with this virus is that the T cell response is really critical. So you would definitely get more T cell response, I think, with either of these. Okay, the easiest to produce in large quantities quickly are these 2 vaccines. And the US has decided to really focus on these. Now, I don't know if you've heard of Operation Warp Speed or Active, but these are 2 government programs where we're basically betting on these vaccines, and we're putting a lot of money into them, assuming that they are going to be found safe and effective. So we're already pouring money into the production and the manufacturing of these vaccines. even before they've been approved or even before they've been shown to be safe. So if none of these work, then we've just lost a lot of money. But if they do work, then we're ahead of the game and we can get people vaccinated as early as January. Okay, so nucleic acid vaccine. So what you do is you take really just a lipid, it's not a virus, it's just a lipid particle, and you have the RNA that you want inside that lipid particle. So it wouldn't be, like if this was the SARS-CoV-2 genome, it would really only be the spike protein or just a little bit of or even a little bit of the spike protein called the receptor binding domain part of the gene that's in there. So if they just have the gene for the spike protein or just the part of the gene for the spike protein in here, this RNA is really easy to produce, and you can literally change it on the fly. So if the RNA of SARS-CoV-2 is changing, you can actually change your vaccine pretty quickly by just typing in a different sequence so it's produced, mass-produced in a different sequence. So with this, this is really just a vehicle for getting the RNA into your cells. So here, instead of with the ChAdOx virus vectored vaccine, this is not a real virus, first of all. And second of all, this is mRNA, not DNA. And so the mRNA comes into your cells. It's a little, it's not as permanent, I guess, if you want to think of it that way. mRNA just lives in your cells for just a short period of time. I can't say lives because it's not alive, but it exists in your cells for a short period of time. You're going to produce the spike protein according to these instructions. And then the way that they've engineered this is that the spike protein is then presented on the outside of immune cells. So the immune cells actually produce this and put it on the outside of their cells, and that tends to give you a better T cell response, hopefully. And that's what they're seeing with the results so far, is that people are getting T cell responses and antibody responses from the mRNA vaccine. Just kind of a funny or interesting trivia, Moderna really is a play on the words mode RNA. So everything that Moderna makes is RNA based. So they make a lot of cancer vaccines, but their mode is RNA, so they're always using RNA. Okay, the last type of vaccine is the subunit vaccine, and that you're just giving people the protein. So either the whole spike protein or part of the spike protein, and you're just injecting that into their bloodstream, and you would be making antibodies against that, because this would be a foreign protein. So the Novavax vaccine is just a subunit vaccine. It's pretty easy to produce this, but it's actually easier to produce mRNA than it is to make proteins in manufacturing. So that's all the types of vaccines. I'm super excited about the mRNA vaccines, but they really haven't been proven to work in any other case. So this is like brand new technology, but I'm excited that they're so easy to make and so easy to produce in vast quantities, which is what we need. And the preliminary results are really good for really both of these. The virus vectored vaccine also has really great results in the phase 2-3 trials. So things are looking pretty good. It's like the best news of this whole epidemic is really that the vaccines that are in development seem to be working.